Welcome to the CortiCare webinar series, which presents topics related to EEG and neurodiagnostics, patient care management, research, and future applications. The FDA and the pharma industry have been developing enrichment strategies that can be used in clinical investigations intended to demonstrate the effectiveness of drug and biological products. From a recent FDA guideline document, enrichment is the prospective use of any patient characteristic to select a study population in which detection of a drug effect, if one is in fact present, is more likely than it would be in an unselected population. Today's webinar presents how EEG can be a critical tool in clinical trials, including in subject enrichment. Presenting today are Drs. Brandon Westover and Donna Murphy. Dr. Westover obtained his PhD in physics, working in information theory and computer vision. He is a board-certified practicing neurologist and clinical neurophysiologist at Massachusetts General Hospital, where he directs the critical care EEG monitoring service and the clinical data animation center. And he serves as director of data science at the McCance Center for Brain Health. His group develops machine learning and artificial intelligence approaches to improve medical care for patients with neurological conditions. And he co-founded Beacon Biosignals. Dr. Murphy completed her MD and PhD and her fellowship in clinical neurophysiology at Baylor College of Medicine and her undergraduate degree at Harvard. She serves as medical and scientific director at CortiCare and continues to support the commercialization of diagnostic algorithms and the growth of the clinical trials unit. Welcome both Dr. Westover and Dr. Murphy.
Jeff's introduction for us. I will just say very briefly that Dr. Brandon Westover has joined us from Beacon Biosignals, has joined us today from in order to co-present with us here at CortiCare. I'm Donna Murphy, the Director of Scientific Affairs for CortiCare, and we wanted to cover subject enrichment in clinical trials using EEG. Let's see if I can advance. I don't know. Okay, great. Thank you, Jeff. Just very briefly, this is the leadership within the clinical trials unit. As I said, I'm the Director of Scientific Affairs. Stephen Johnson is in charge of clinical trials business, and he has extensive experience in that domain. Robin Shabano has joined us in the last year as the operations manager for the CTU, and Tim Innes is overall at CortiCare in charge of business development as the executive vice president. So I just wanted to acknowledge those folks. And the objectives of today's talk, I wanted to review very briefly. So early phase clinical trials of compounds that may compromise or otherwise change brain function warrant monitoring or analysis for subject safety and efficacy. EEG can be very helpful to those ends. So this webinar aspires to cover rational design, strengths and limitations of EEG, as well as standardization of EEG monitoring and analyses in clinical trials. And specifically, we'd like you to leave by the end of the webinar with the ability to identify the challenges in CNS drug development mitigated by EEG. We'd like you to be able to define the 3 strategies for subject enrichment in clinical trials, to consider the operational and cost constraints, as well as the scientific trade-offs of various EEG trial designs and different elements that you may or may not want to include in your EEG trials as sponsors and as CROs involved in trial design, and then also to characterize the limitations of EEG in clinical trials. So just a little bit of background here on CNS drug development, which can be very, very challenging. In general, it's important to understand the context of drug development. For those of us who are not aware, fewer than 1% of compounds make it past preclinical testing or testing in animals. Greater than 90% of those that make it past preclinical testing will actually drop out in phase 1, which is first in human clinical trials. Those typically are in ostensibly normal or clinically normal human subjects who pass screens on being normal. It takes over a decade and spend about $1 billion to take a compound from bench to bedside. So you can imagine it's an enormous investment. It's an enormous investment with a lot of attrition, right? And so it becomes very inefficient, and CNS trials outcomes are actually even worse than this. And just to give you an example, other than in multiple sclerosis, disease-modifying treatments and chronic neurodegenerative disorders, the dropout is 100%. So we haven't had really much success in neurodegenerative disease in taking compounds from bench to bedside. So these challenges then make it very difficult for sponsors, and they have, in large part, many sponsors have discontinued their CNS drug pipelines altogether. And this is despite the fact that neurological and psychiatric diseases continue to generate enormous health and economic costs. So this slide is just to give us a little bit of background. I have a couple of slides here about subject enrichment that can help to de-risk the process of developing these kinds of drugs, right? And these would be both for drugs that have CNS therapeutic purpose, and also for drugs that actually are not targeting the CNS, but penetrate the blood-brain barrier and may have may cause functional changes in the brain that may not be desirable. So for subject enrichment, EEG, it's very helpful in this regard because it will help us to avoid adverse CNS outcomes with the CNS penetrating drugs designated for other therapeutic purposes, as well as for CNS drug development. If the concern is, for instance, seizure potential of a drug, right? Screening for baseline epileptiform discharges as seen here on the left, the sharp wave spike and wave discharges that can be identified by expert human reviewers. Can enrich for subjects who are truly normal, right? So people who are clinically or ostensibly normal may not in fact be normal because they have clinically silent abnormalities in the brain. Anecdotally, across hundreds of studies at this point in Corticare, we've noted 15 to 20%, so a very high percentage of clinically normal subjects exhibit EEG abnormalities of some kind. Still, the epileptiform activity at baseline is pretty rare, but on-study seizures or seizures in patients epileptiform discharges in patients who have been exposed to a compound with a clinically inapparent vulnerability where EEG has not been used to exclude them can put those subjects at unnecessary risk. And also it could prematurely abort the entire clinical trial, right? If you have a bad outcome during the trial because you haven't appropriately screened. So quantitative and qualitative EEG assessments, in this case, we first talked about qualitative assessments with the human expert review on the left, and then on the right, quantitative assessments using algorithmic identification of seizures and epileptiform discharges are also possible. And the picture on the right is alluding to that. I also wanted to mention that the kind of analysis that can be performed can be either real-time monitoring using, for instance, at Corticare, we have an expert network of registered EEG technologists who can monitor subjects in real time who are exposed to a drug. It can also be that we do retrospective review by our experts, our board-certified clinical neurophysiologists who are concentrating in EEG or epileptologists who are board-certified. The next slide I wanted to talk about here is also about subject enrichment, helping to de-risk this process. And this is more specifically about quantitative EEG, which we won't spend a ton of time on. But this is just to show you that we can, often this is retrospective review, we can retrospectively improve trial efficiency by using quantitative EEG. One useful example of this is in performing a pharmacodynamic analysis in early stage trials to inform the time window over which a drug is studied to capture only when it's functionally relevant in the brain, and also to inform us how that drug should be dosed in later stage trials. To give you some background on quantitative EEG, this is a really useful biomarker and it still is being developed. It objectively measures the pharmacological response or the pharmacodynamic response of a biological process that can be quantified, is reproducible within subjects, and also you can look across a group as well to see the patterns across the group. In the EEG, it can be used in healthy subjects without reference to a specific therapeutic indication, as seen here. This data is actually from healthy, or quote unquote healthy human subjects. And it also can be used in patients with specific illnesses, particularly where EEG measures correlate with disease prognosis or therapeutic responses. So here, just briefly, these pictures are of analysis that was done on the power spectrum of the EEG with eyes open and eyes closed. And we were examining frontal alpha activity as well as frontal beta 1 activity. And you can see that on the left, the alpha activity does not actually parse out by the doses or the dosing cohorts, right? The 1 through 6 there in the different colors. But they do parse if you're looking at beta 1 activity on day 1. On day 14, everything actually is, you can differentiate all of the different cohorts there. So this is important to know, right? Because then you may want to use beta 1 activity then if you're looking at the changes that can be seen on a shorter time scale. Okay, EEG enrichment, very briefly, is there are 3 kinds of enrichment. And there's a really wonderful document by the FDA that explores all of the different kinds of enrichment in both safety and efficacy trials. And we're just focusing the enrichment around what EEG can provide here. So I've just provided some examples in these 3 different domains that the FDA outlines, and that is in decreasing variability, in prognostic enrichment, and also in predictive enrichment. So for decreasing variability in safety EEGs, right, we were talking earlier about enriching for people who are truly normal. So they maybe have clinically silent abnormalities that can be detected if you do an EEG. You might see epileptiform discharges, for instance, and you would not want to include those subjects when you're screening a compound for that potentially has a seizure risk associated with it, right? So you don't want people who already have vulnerability to seizing. In other words, you'd want to exclude them from your trial, and that would decrease the variable response that you might have. And that, is in the best interest of that subject. It's also in the best interest of the trial to screen those folks out. Prognostic enrichment and predictive enrichment, I think both of the examples that I provide here use algorithmic EEG or quantitative EEG. So in the first example for prognostic enrichment, we can use a spike and seizure detection algorithm for event-driven or continuous endpoints to measure absolute effect of an anti-seizure medication. And this is an example of prognostic enrichment and how you can use EEG in that way. The event-driven enrichment is going to, or endpoint is going to be the presence or absence of, for instance, a seizure. And continuous endpoint would be worsening of seizures as an example, or epileptiform discharges. Predictive enrichment, the example that I provide here for that is use of quantitative EEG to select for subjects whose functional responses to specific drug doses will help you to predict whether they're going to also respond to that treatment clinically, right? So that's something that you'd like to be able to predict reliably, and the quantitative EEG gives us some promise of that. Great, so EEG trial design considerations. I just wanted to say briefly that activation procedures such as photic stimulation and hyperventilation can be used in order to elicit generalized epileptiform abnormalities. They also have been shown to highlight some focal abnormalities which may be elicited as well from those procedures. The operational constraint here is that you'd have to have a photic strobe and usually some expert who's administering the activation procedures in person in order to perform those. So that is something to keep in mind when you're designing your EEG trial and whether you want to actually include those procedures. EEG caps versus the traditional 1020 application performed by somebody who is expert in applying that whole set of electrodes. EEG caps are obviously a lot more efficient, but they often will miss abnormalities. They will have more artifacts than the traditional 1020 application. So those are the trade-offs that you have to kind of contend with there. The baseline and on-study EEG, when you're thinking about how much of the baseline to get, whether to capture a baseline. Often people think that we can just do a very abbreviated baseline, a 30-minute screen with or without activation procedures, and then do maybe a more extended on-study EEG to examine the whole time over which you have maximal presence of that drug in the system, particularly in the brain, right? But the problem with this is that you're comparing apples to oranges in this case. And so you want ideally the same amount of time over which you're comparing the presence of, as an example, epileptiform discharges or seizures prior to and after the drug dosing, right? And a really good piece of data that emphasizes this point, the importance of actually doing time-matched and duration-matched comparisons, is this picture out of a paper that I have linked actually in this presentation, which hopefully we can make available to you all. And this actually shows on the x-axis the time of day. The gray areas are indicating what is the nighttime, so typical sleeping hours. And you can see there's this kind of diurnal variation in the presence of spikes and seizures, and it depends on the individual. So each of these plots is an individual person whose EEG was observed over this time interval. So that's really important, right? It means that you may elicit seizures or epileptiform discharges with a compound, but if somebody already had that vulnerability, you can't necessarily say it's the compound that's at fault. It could be that they just naturally were going to declare themselves in the period studied after dosing. Particularly when you're studying a larger interval of time, you're likelier to capture those abnormalities. So you want to make sure that your duration matched, and also time of day matched because of the circadian variation. Next slide, please. Great. And so I just wanted to end with the CortiCare Beacon partnership, which we're really excited about. This is kind of a preferred partnership. I think each also work with other partners. But CortiCare, I just wanted to emphasize some of the unique and also overlapping services that we have. And Dr. Westover is welcome to also expound upon what Beacon does beyond what I've provided here. CortiCare is involved in EEG trial design consultation with sponsors. We have a whole process in place, and also the expert knowledge and the networks that we've developed also through the clinical side of our business at CortiCare to support robust data collection, reliable, reproducible data collection, data transfer, storage, and cloud access. We also have provided to our sponsors and CROs. We have also designed custom quantitative EEG analyses for the needs of our sponsors, and have also provided guidance on what exactly we should be examining per the literature and what we understand about the theoretical the theoretical understanding we have about a compound and that class of compounds. And then we also have an expert board certified network of central EEG readers. Beacon also has a fantastic board certified network of central EEG readers, and they have a proprietary algorithmic spike and seizure detection option that we have been utilizing in partnership with them that has been very, very helpful as an adjunct to our expert clinical review by our human readers. And then finally, they also have been engaged in quantitative EEG deployment. So I'm going to pass it over now to Dr. Westover to finish us off with some of the limitations of EEG in clinical trials.
Thanks, Donna. So I want to take you a little bit kind of deep into some of the considerations around safety EEG reading. In particular, detecting epileptiform abnormalities. I'm going to really be talking about epileptic spikes and in clinical trials. So I'll take you through what are some of the issues that can come up. By the end of this, you'll understand some of the most effective ways to deal with these problems. And then we'll, if you follow these practices, then you get valid safety data from clinical trials. Before I kind of launch into the material, I just want to acknowledge I'm going to be talking about some of the work that I published with my academic hat on, and so these are the stars of the academic work that I'll be talking about on inter-rater reliability and automation of detecting epileptiform abnormalities, and then I'm privileged to be part of a team of a diverse team, actually, of computer scientists, neuroscientists, neurologists, and then AI engineers at Beacon Biosignals, where we take things that my group and others have published in the literature, as well as developing new things. So this quantitative EEG that Donna is talking about, we developed that, and we develop kind of standard things so that we can do them at scale, and then also new things as needed for specific clinical trials. But today I'll really focus on epileptic spike detection. So let me just first talk through some of the problems. And so the first problem is the problem of false negatives, right? So missing events that are there. And I'm going to just tell a real story from one of our clients. So this was actually a company that we worked with, the Beacon worked with, who had purchased actually a new drug program. They wanted to develop a drug, and this was it's a $300 million deal approximately. And what they found when they were starting to ramp up is that in some of the data that they had from earlier work, from before they acquired this portfolio, there were a couple of cases that were problematic. In fact, raised the possibility that the drug might actually put patients at risk for seizures. And so in one of the cases that I'm showing you here, this is actually from a pre-dosing period. But actually, this is pretty clearly an epileptiform discharge, right? You can see that it's kind of bifrontal. It's a little bit more prominent on the left side, but it's probably the kind of spike and wave discharge that you'll see in a patient with a genetic predisposition to seizures and idiopathic or genetic, generalized epilepsy. But this had not been seen. What had been seen was several examples of epileptiform discharges that looked much the same in the post-dosing period, right? So this was a missed event. And why was it missed, right? It's pretty obvious when you look at it here. But the issue was that this data was 24 hours long, and there were only 4 of these discharges in that data. And so it's literally like looking for a needle in a haystack. And it's not surprising, right, that this sort of thing would be missed, especially given what I'm going to show you in a couple of slides about interrater variability. So needless to say, we were able to review this with some of the techniques and measures that I'll show you in a bit and find these. And so that having good sensitivity is actually essential to being able to do safety EEG reading. In this particular case, this specific case was actually able to be removed from suspicion because the patient had the same discharges before and after. They probably had a trait that they just never knew about. And patients with known epilepsy who'd had seizures or convulsions were excluded from the trial, but this is probably a patient walking around with a risk that they just didn't know about. The next problem is the problem of false positives. And so I'll show you another. This is also from the same set of examples from this one drug study. And this was a case where the EEG technologist who read the study as part of this kind of clinical research organization that provided EEG reading for the company had said that, well, a seizure occurred at this time. And this was in the post-dosing period. And so again, this is one of the cases we were asked to review to decide if this was a real concern or not. And I think you can see the patient's awake initially for this first 2/3 of this little segment. And then something happens, right? The EEG changes dramatically. There's all this slowing going on. And so this is what was called a seizure. The patient did become unresponsive. But if you look carefully, there's what was missed here is the EKG, right? The EKG, there's this pretty severe bradycardia that coincides with this change in the EEG. There's really no epileptiform activity in the EEG. It's not really rhythmic. There's no spikes or sharp waves in there. This is a continuation of it. It's still this bradycardia continuing on. And then as the EKG normalizes, you can see the patient actually regains consciousness and their EEG normalizes as well. All right. So this is one example of a misclassification or a false positive. In this case, a false positive seizure. And this is another case that we were able to really remove from suspicion by more carefully examining the data. But again, it's difficult when you have really a lot of data to review, and then some problems with inter-rater reliability that I'll talk about in a moment. The last problem I'll talk about is it's really kind of similar to the previous two problems, but I think it has a different flavor. And this is the problem of information overload. So to illustrate what I mean, this is an example from a different client that we have. In this particular case, the drug being developed is being tested in children that have very frequent epileptiform discharges. This is one example of a child with hypsarrhythmia, and you can see we've kind of annotated using an automated method that we have all these different epileptiform discharges. And in fact, in this, the drug, the hope is that this drug will decrease the frequency of these epileptiform discharges. It's not known yet how much that will be, but the desire is not just to make a yes or no, this patient has epileptiform discharges or not. It's to say, what's the percent reduction that this drug achieves, in a given patient. And in one of these studies, in just an hour, you'll typically find 1,000 or more of these epileptiform discharges. And manually annotating these the way that we do conventionally is just impossible, or very tiresome in any case, pretty much impossible. Here's another example. This is actually from a paper, but this is similar to to the trial I was just talking about. It gives you a sense of what kinds of things we need to do in some analyses. So this is these are patients with two different conditions. You can think of these as different drug conditions. And so imagine over a 15-day period, you have to say which group has more, and actually not just which group has more, but how many more epileptiform discharges than the other group. And so here, these are subjects on the y-axis. There's time. It goes up to 15 days of continuous data in some of these cases. And the black indicators are actually color-coded. Well, the intensity of the black, how black it is, indicates the rate, the number of epileptiform discharges per hour. And this is the kind of analysis that we've been asked to do in other trials. So again, this is not really feasible manually. There will be this information overload problem, and humans doing this will just be inaccurate. So I'm going to tell you how to understand, these problems, why they happen in a little more depth, and then this will help us understand how to address them. To introduce this, I just want to actually motivate it with a story, personal story. So this is my daughter a few years ago. And she actually one morning had a convulsion out of the blue. It was terrifying. Thankfully, she was okay. But of course, the question we had in our minds after she was fine was, will this happen again, right? In the end, it boiled down to a test result. She got an EEG, and depending on if we found something abnormal there, we were going to be on one course of prescribing a treatment. And if there was nothing, if it was normal, then this isolated event with no other, indications of high risk for seizures, we would have just watched and waited. And so this brings up the importance of inter-rater reliability, right? So it matters a lot if we show the same data to different experts, because you don't know which expert you're going to get. If you're me here, you want it to be the same either way. Similarly, if you're running a clinical trial, you don't want it to depend on which EEG experts you get, right? Whether somebody's going to be found to have an abnormality or not. You want it to really represent something true in the data. And of course, if you're a participant in a trial, you want to know for sure if you have something abnormal in your EEG that you're, properly excluded from the trial. You're not exposed to undue risk. So this is very important and ultimately, economically important as well as for safety, especially in drug trials. So I'm going to talk a bit about an academic study that we did, my research group did, to understand the problem of inter-rater reliability. And so this was a study where we had 8 epileptologists, so subspecialty trained physicians. These are gold standard experts, rate over 13,000 epileptiform discharges are candidate epileptiform discharges. And so this is an example of the type of data they had to look at. So here the question is, between these two red lines, is there an epileptiform discharge? And we got a yes or no vote from these eight experts here. 1 out of 8 people said yes. I think I would have said no here. Here's one where 4 out of 8 said yes, and I would be somebody who says, yeah, probably there is one here, down here at T6. And then here's one where 8 out of 8 agree that there's an epileptiform discharge, and this one's very, very clear up here. So this gives you some sense of the variability between different patients. Right? These can be lots of different shapes and locations, and in the end we got data that looks like this. Right? And so what we did is we binned those 13,000 examples based on how many votes they received. So that's the x-axis here. And then on the y-axis, we're making a curve for each of the 8 experts who scored those examples. And the curve tells you what fraction of the examples that expert voted for. So here's this case up here with is an example of an overcaller, somebody who when for all the 2 out of 8 spikes, this person votes for most of them, so they overcall relative to the group. And then here's someone who's very much an undercaller, and then we have some people near the middle. You get kind of similar results if you do at the whole EEG level. So here, what we're doing on the right is we're saying what fraction of if you either vote any spikes in an EEG or none, and you can see still we have overcallers and undercallers. All right, so this is a bit alarming, maybe if you're me and you know that your colleagues, one colleague might find the EEG abnormal and the other one might not. What does that say about the ability to provide the right treatment? And we all know that there are problems with falsely positive diagnoses and misdiagnoses, in part based on this variability in EEG reading. So now I want to go dig a little deeper and understand why this happens. Right. So we've done a follow-up study that's I'll just mention a little bit here. So we got on the left, I'm showing you those calibration curves. The black curves are the same data I just showed you. We collected data from an additional series of experts, and these experts were each asked to annotate 1,000 epileptiform discharges. So the black curves are from people who did 13,000. The blue ones from 1,000 each, and we have some some experienced but not expert people, and then some novices. And what you can see over on the right-hand side is if we just consider the correct answer to be, if the original 8 experts, we're calling them super 8 here, marked at least half, if at least half of them agreed something was an epileptiform discharge, we'll call that, the correct answer is yes, otherwise no. And so with the correct answers defined that way, we can define for each person a false positive rate and a sensitivity. And what you can see is the experts, the blue and the black dots, are kind of up and to the left on this ROC type of plot of the novices, the red ones, right? So those are generally down and to the right. So let me come back to that in a moment. And then the experienced but not expert people are in between, as you would expect. So we were able to find essentially that I won't go into all the details, but the following model describes where the variability comes from quite accurately. So the model is when one of these experts or non-experts sees the data, right? So imagine this is part of an EEG, a candidate epileptiform discharge. They see it, but they add to it in their mind essentially some noise or uncertainty. And so what they really see in their mind's eye when they make a decision is this noisy observation. And then they also compare this to a threshold, which creates a bias, right? So different people will have different thresholds. And when the noisy observation seems to have enough evidence relative to that threshold, the person will call it a spike and otherwise not a spike. Right? And so what we find when we look at these groups is that in general, the experts seem to have a lot less noise when we estimate how much the amount of noise in the system. It's low, and the novices tend to have a lot more noise. This means that effectively what's going on is that people are operating on these different ROC curves, right? So the amount of noise determines which ROC curve you're on. The experts are on this better curve up and to the left than the novices. But the threshold, this internal threshold, what level of evidence you require to say yes, that is sort of a matter of personal preference in practice. And that's where most of this variability among experts comes from. It's not that they perceive the evidence differently. They more or less see the same underlying probability, but depending on what threshold they're using, they vary. Right? And so this is an addressable problem, right? It's a much more difficult problem if they were sort of some were good and some were not good. Right? So that's refreshing. And so I'm going to suggest a couple of things that can help combat some of that variation. So one is if you're stuck using human readers only, right, then you can at least be systematic about the way you read EEGs. The ideal situation is not to be using only humans, but to actually be using AI in combination with humans, which I'll talk about in a minute. But first of all, either way, you need to be systematic when there's difficult cases. So I'm going to tell you a little bit about some of this systematic approach. If you want to learn more, I'll just shout out to Fabio Nascimento, who's organized these pretty neat podcasts, and you can search for these on YouTube, and there's educational videos about sort of how to read EEGs and in particular about how to identify epileptiform discharges. But just very briefly, there's a systematic approach that we've adopted. It's from the ILAE and IFCN recommendations about what characteristics you should use when you're judging a waveform to decide if it's epileptiform or not. And so there's 6 criteria. The first one is is the shape, is it spiky or sharp? And so this is an example. Obviously this one is spiky. The second thing is the duration of that wave different from the waves in the neighboring background. So here would be this duration is much longer than the spike, so they would pass that test. The third one is you look at the slope going up and down of this spiky component, and if the slopes are different, that's evidence in favor. The fourth thing is the presence of a slow wave, and of course, not every epileptiform discharge has one, but when it does have a clear aftergoing slow wave, that's evidence in favor. The fifth thing is, does it stand out from the background? Does it disrupt the background? And then the sixth thing is whether it has a physiological field, right? And so these things epileptologists learn to evaluate. They don't always apply them systematically. They kind of learn to sort of have a reflex response, having seen thousands of examples. But when there's a difficult one, something that's sort of ambiguous in between, it helps to really go through this systematic process and make a decision about each criterion. The recommended kind of threshold is that something should have at least 4 out of 6 or more of these characteristics to be called an epileptiform discharge. And this rule works pretty well. So here's an example that clearly kind of meets that criteria. So it has a spiky morphology. Its duration, if you look at it carefully, is different from these small waves in the background. The slope, I think, is a little steeper going up than down. It has a clear slow wave. It disrupts the background. And the physiological field is physiological. This looks like something that comes from the brain. So again, with some training, epileptologists should all be able to go through those things and evaluate them. This is an example of a difficult case. At least we found it pretty difficult. And this is actually this is a case that actually ended up screening someone out of a trial. But if you look carefully, the thing in question is this little transient up at T3, and it's pretty low voltage, and it seems to have a little bit of a slow wave, but it's not very well formed. But if you go through these, so I think if you showed this to people and just asked them, just tell me your gut instinct, you'd probably get somewhere near 50-50. It could be a little more or a little less. Of course, this matters a lot if this is a patient in a trial or a patient getting a diagnosis. So, just going through the criteria to be disciplined, it is sharp and spiky, at least in my judgment. I think the wave duration is not really so different from the neighboring waves, and so I didn't give it credit for that. I think this looks more triangular, so the slope going up and down looks about the same. But I thought it did have, although it's not very well formed, an aftergoing slow wave. I think it disrupts the background, and it has a field that's reasonable at F7 and T3. So again, this is an example a difficult case that you can at least resolve in a disciplined way by using these criteria. All right. So, I won't really go into this much, but of course, one way to combat variability, which was suggested by the plot that I showed you, was to have multiple readers. Now, how many readers do you need? Well, unfortunately, it's probably a fairly large number. In some of the research we've done, we find that somewhere around 7 or 8 is actually ideal. And that's pretty difficult to ever achieve in practice, just kind of too expensive. But only around that level do you actually start to see that, for example, there's a very close correlation. If you take the consensus among sort of 7 or 8 readers, you start to see a very close correlation with objective features in the signals. Again, how do you get there? Well, actually, the way to get to the level of multiple raters without actually employing 8 people to read each EEG is to use augmented kind of intelligence, so artificial intelligence together with the human reading. This is just to remind you about the reason multiple readers is a good idea. You average out people's idiosyncrasies. So let me just talk now about the AI part of this, and this is what I'm most excited about. So the idea of automating the detection of epileptiform discharges, maybe it seems like a pretty simple task, and certainly a lot of people have tried, even going back to the 70s. But until just recently, this had never been done very well because it's difficult to do well, I think. And I just want to hold up one example of what I think was really one of the best studies until Dr. Jing's study. But just to illustrate, even among the best studies, what have been the problems in the literature to date. And so this is a study from a company actually that makes software that, among other things, detects epileptiform discharges. This is from the Persist 13 version of the software, and they published a study where they made this very bold claim that their algorithm was able to essentially, if you read it, it reads as if it can replace people. Right? It passes the Turing test, meaning that it can read or detect spikes just as well as experts. But the demonstration really, I did not find it all convincing. So they evaluated the algorithm on 40 patients, which is very small relative to the range of different types of spikes or abnormalities that we see in EEGs when you look across all comers in the population, say in a typical EEG lab, or for that matter in patients undergoing clinical trials. These were also not just any patients, though. They were patients with coming in for the most part to be evaluated for epilepsy surgery or in an epilepsy monitoring unit. So maybe not the typical patients who would come into a lab, most of whom never come to the EMU. And then finally, the reference standard in this case was not physicians, and so we don't really, this can't be considered a gold standard. So we set out to try to create a general purpose spike detector. We sometimes call this SpikeNet. So in the academic version that we published, this I think we called it SpikeNet. But what we did was we collected over 1,000 EEGs from all comers, and we managed to get people to annotate over 88,000 candidate epileptiform discharges. We were able to cluster those down into 13,000 small clusters. So many of these things look the same as each other. And so in the end, it was 13,000 that we got 8 experts to annotate. And so the spike detector that we developed is based on this. The inter-rater agreement studies that I mentioned earlier were also based on this. And so my claim is that this is much closer to a general purpose spike detector than anything that has been published previously. And this is a neural network that we developed. And so here's the ROC curve. If you just consider the answers to be yes and no, which is typically the task that we have people perform, right? We don't give probabilities. We say yes or no to the spikes. And so you can get a much better ROC curve than the algorithm that was published with the claim that it passed the Turing test. You also see better calibration. And so this blue curve over here on the right is the algorithm. And the thing that's really kind of nice, I think, in using this for something like this for clinical trials is that what you can see is that although, again, it's impractical to have 8 people reading one EEG, this algorithm does essentially what you would, what you get, right? If you had 8 people kind of try to decide, right? You get something like the average among those 8 people. It really gets rid of the noise in the process. And this is an example of the type of output you can get, so you can annotate every epileptiform discharge. The computer doesn't get tired. It doesn't, sort of change the way it's doing things. So you will get consistency as well as this accuracy and calibration. And so these are just some other examples of patients with a lot of spikes. Now, the results I showed you before are published, and those are academic. At Beacon, we've developed a version of this that goes way beyond what we've done before. It has a better ROC curve, and it has some other fancy properties that I won't go into here. But this can now be used in clinical trials at scale and very quickly. So that makes reading these EEGs much easier. So I'll just sort of end it here with some take-home points. I think hopefully Donna and I both convinced you of the reasons that EEG can be very valuable in enriching clinical trials. In particular, for my part, enriching safety trials, right, with people who really do belong in the study, and then for screening out people who would be at risk if they participated. I've shown you what some of the problems are, these false positives, false negatives, too much information in trials where you want to really quantify the spike rate or the burden over a long period of time. And I've told you a bit about why inter-rater reliability problems are present when you have, even though you have well-trained experts. And I've told you a bit about how to overcome those problems by using a systematic approach, multiple readers when you can, although again, it's hard to have enough readers to really overcome it. But with artificial intelligence or algorithms that are able to learn from large numbers of examples annotated by large numbers of experts, you can really essentially get what you would have if you had the luxury of having, say, 8 or more EEG readers. And you can do this, of course, much faster, and then reduce the problem of false positives, false negatives, and information overload. And so following some of these practices will we believe give you better results from your clinical trials. So I will stop here. And
I just wanted to say, just to kind of give a plug to the other arms of what work QuartiCare does. Since Jeff, I'm not sure that everybody was able to hear you when you were introducing us in the beginning. Everything I think that we've discussed here is of great utility also in standardizing reading in the clinical realm. So for those who've joined us who are clinical neurophysiologists or epileptologists, and also technologists, honestly, I think some of these strategies that Dr. Westover has provided on how you can systematically review what may or may not be an epileptiform discharge for some better agreement between readers, that this is something you can apply even in clinical work and not just within the clinical trials domain. So hopefully that's been of utility to everyone, even for those of us who are not in clinical trials.
Well, I want to thank both of you for this very engaging topic. I think it's one of those components that advances the science of EEG and certainly would enhance the quality of clinical trials going forward. So again, appreciate it, and we look forward to seeing all of you again in July. Take care.
Wonderful. Thank you.
Thanks.
Goodbye.